evening, everyone. I am Kushal Pramanik, a student of UG 4th SEM of Chemistry Department, Durgapur Government College. For today's presentation, my topic is bioactivity of Geraniol and Avobenzone through computation. And contents of the project are introduction, objectives of the project, methodologies have used, main findings, and conclusion with some references. So, So before going to the main topic, let's see what is bioactivity. So the term bioactive is an alternative term of biological active. Bioactivity is defined by obtaining specific effects after exposure to a particular substance. These effects may include tissue uptake, metabolism, or physiological response. So, in easy words, bioactivity of a compound is the specific effect or influence of the compound in our body and physiological process. So these are my chosen compounds. First one is geraniol. This is the structure of geraniol and the UPAC name is 2E37 dimethyl octa 26 diene 1-ol. Geraniol found in essence of some flower like citronella, rose, and Cymbopogon citrus. And geraniol has a very pleasant smell, so it used in perfumes and the scent such as peach, raspberry, grapefruit, red apple, palm, lime, orange, lemon, etc. And second one is avobenzone. This is the structure of avobenzone, and this is the IUPAC name of avobenzone 3 4 tert butyl phenyl 1 4 methoxypentyl propan 1 3 dione. And avobenzone is dibenzoyl methane derivative. It is soluble in oil ingredients, and avobenzone absorbs ultraviolet light over wider range of wavelength. So it used in sunscreen products to absorb the full spectrum of UVA rays. Then these are the objectives of the project. First is we have used the ChemDraw software and Pass Online website to analyze the bioactivity of the chosen compound. Then second one is we have done the literature survey of the compound and explained them in this project. And these are the bioactivities of geraniol. First one is lipid metabolism regulator, then antimicrobial, then anti-tumor, and the last one is anti-inflammatory. And the bioactivities of avobenzone are carminative, antineurotic, cholesterol antagonist, gonadotropin antagonist, and phobia disorder treatment. These are the methodologies I have used. In step 1, I have used ChemDraw software to draw the structure of my chosen compounds and to make the mole file. In step 2, I have registered in Pass Online and get the prediction about the bioactivities of the compound by submitting the mole file. And in step 3, bioactivities of the chosen compounds are noted. So these are the main findings. Bioactivity is chosen for the geraniol from Pass Online website. First one is lipid metabolism regulator. Lipid metabolism is the synthesis and degradation of lipid in cells, involving the breakdown and storage of fats for energy. We propose that geraniol could be a potential agent for the treatment of hyperlipidemia that could contribute to the prevention of cardiovascular diseases. The aim of the present study was to advance our understanding of its mechanism of action on cholesterol and triglyceride metabolism. So the hyperlipidemia is the condition when there are high levels of fat particles or rather than lipids in our blood. Geraniol has a potential to treat hyperlipidemia and prevent several cardiovascular diseases, and it normalize our blood circulation. Second one is antimicrobial. An extensive review paper on the antimicrobial properties of geraniol has recently been published. Such an activity of this compound may result mainly from its non-polar character, which it may disrupt the lipid structure of the microorganism's cell membrane. Interact with these components make it more permeable also for the compounds, example, antibiotics. Geraniol can also penetrate the cell interior and limit the growth of the microorganism by binding the intracellular places of key importance for its survival. So, in easy words, the substance that antimicrobes are a substance that kills microorganisms such as bacteria or mold, or stop them from growing and causing diseases. So in this case, a geraniol disrupts the lipid structure of the microorganism's cell membrane, and it helps antibiotic to penetrate by making it more permeable. And geraniol can also stop the growth of microorganism. Third one is anti-tumor. Geraniol displays anti-cancer activity against many human cancers in both in vitro and in vivo studies. Although many publications have been published about its anti-cancer activity, in mechanism of geraniol molar activity has not been fully explained yet. So the anti-tumor or anti-cancer activity of geraniol is confirmed by vitro and vivo studies. Vitro means in glass, so the study experiment in test tube are vitro study, and the vivo means in live, so the experiment in human or animal is called vivo study, but the mechanism of the activity is still not been explained. Fourth one is anti-inflammatory. Geraniol significantly reduces the expression and release of cellular pro-inflammatory factors TNF alpha, IL-1 beta, IL-8, and nitric oxide, accompanied by a significant upregulation of geraniol expression of anti-inflammatory cytokinin IL-10 in LPS induced WBCs compared to non-treated cells. It demonstrates a much stronger inhibition potential than diclofenac in terms of inflammation inhibition. Then bioactivity is chosen for Avobenzone from Pass Online website. First one is carminative. Carminative compounds are those which contain volatile compounds that have traditionally been used for their pharmacological properties on benefiting human health. Primarily, avobenzone have therapeutic effects in supporting skin health and influencing overall well-being. So avobenzone also work as carminative. Carminative is a herb or a preparation which prevent formation of gas in our gastrointestinal tract, and it help infants in help their treatment of colic disease. And second one is antineurotic. So antineurotic, like avobenzone, also known as antipsychotic compound, are used to treat and manage symptoms of many psychiatric disorders. Avobenzone typically work by affecting dopamine and sometimes serotonin levels in our brain to decrease psychosis symptoms. Dopamine and serotonin are both neurotransmitters, small molecules that transmit information across nerve cells and affects many major body systems like cardiovascular and nervous system. Third one is cholesterol antagonist. Cholesterol antagonists are a class of compounds that prevents the uptake of cholesterol from the small intestine into the circulatory system. Avobenzone block the uptake of micellar cholesterol, thereby reducing the incorporation of cholesterol ester into cyclomicron particles. By reducing the cholesterol contents in chylomicron remnants, cholesterol absorption inhibitors effectively reduce the amount of cholesterol that is delivered back to the liver. Fourth one is gonadotropin antagonist. So gonadotropin hormone causes in pituitary gland in the brain to secrete luteinizing hormone and follicle stimulating hormone. So pharmacological management of endometriosis significantly increases the importance either as a first line treatment or as a complementary therapy after surgery. Yet current research on antagonists of the gonadotropin like avobenzone has revealed their potential benefits in endometriosis treatment. Fifth one is phobia disorder treatment. Treating phobias can involve mental health therapy and medication, or a combination of both two. And the mental health therapy, the formal term is psychotherapy. The treatment can depends on the type of phobia you experience and factors related to your lifestyle, routine and preferences. A healthcare provider can tell you more about the possible treatments that can help you and which they recommended. Lastly, Avobenzone have a very important role in treatment of phobia. Then conclusion, by using computational study, I was able to find out that the following biological activities of my chosen compounds, geraniol and avobenzone. It is now being clear that a bioactive compounds is a compound which has the capability to interact with one or more compounds of living tissue. Then these are some references I have used for preparing my presentation. And at the end, I would like to thank our HOD, Dr. Snigdha Chandu ma'am and Dr. Nivedita Acharya ma'am for arranging this amazing course. And also, I like to specially thank Oshmita Mondoldi and Borsharidi for guiding us through the whole course. Thank you.
Yeah, thank you, Kushal. It was really nice. We have Principal